Good afternoon. Diane or Paula, are you on the call? How about any of our speakers for today? Tessa, Emily, Jenny, are you on the call?
Sean, this is Diane and Paula. You got unmuted there.
Oh, no problem. Morning or good afternoon, guys.
Me too.
Can you confirm which screen you're seeing of mine? I've got two monitors here. Is it the
I
main
see
one or is it the briefing?
the first slide.
Okay, and not in presentation mode, you just see the full slide.
Correct.
Okay, then I think we're all set to go.
Yep, and I showed Tessa's logged in, but they're muted right now, so they might not be at the desk. So.
And no word from Jenny yet?
Not yet.
Okay.
Hello, good afternoon. This is Jenny Liu from USP.
Hello, Jenny.
Hi, Jenny.
Hi, Paula and Sean. Paula.
Perfect. So we're all here.
Okay, great.
Jenny, just so I get it right, could you pronounce your last name? Is it Liu? Is that right?
Ah, correct. Perfect.
Very good. Perfect. Just didn't want to get it wrong.
No problem.
And then Tessa, do you know if you guys are able to unmute and check your audio? You might still be working on it.
So I have a question. Yeah, so I have a question. For the questions, you will show it on the chat section, or where can I see it? Or the Q&A?
So it would come through on the Q&A. Actually, it could be either, I guess. And last time, what we did is we went through all the presentations, and then people were able to use that during the presentations or after to ask questions.
Mm-hmm. So the Q&A will be at last?
Correct.
Okay. So, and they will type their questions in the Q&A box, and we can see that?
Correct. And we'll also read them off too before we answer them. So.
Okay, great. Thank you.
Uh-huh.
Diane, I don't think we've heard from the Bion Sciences group yet. I had sent them a message here in the chat as well if they could confirm their audio, but if they're able to hear us, I don't think we're able to hear them yet. Did you catch that, Diane?
I see Tessa's in the panelist, but I don't hear any audio from her.
I just sent them a quick email to see if they can get their audio going.
Diane, can you hear me?
Yes, I can.
Okay. Have you heard anything from Emily or Tessa yet?
No, I just sent them an email to check their audio. I see that they're logged in. It's just looks like it's muted. So.
Okay.
And then for everyone who's joining us, welcome to the second webinar that we've got here. We'll get started here just in just about 5 minutes. Let's some more people join us. So thank you for your time.
She's on the phone and asking if she can get to you directly. You can transfer her in.
Hey, Tess, I think we can hear you now.
Yeah, sorry, I had myself on mute.
Oh, no problem. Okay. As long as we get that figured out, we're good to go.
But I hadn't heard anything either. So if anybody was talking to me.
We were just checking for audio. So
Oh,
as long
okay.
as you can hear us now, you're good.
Okay, sounds good.
Great. Thanks. We'll start in just a few minutes.
Okay.
For everyone just joining the call, we'll get started here in just a few minutes. Let some more of the attendees get logged in. Thank you. So hello, everyone. It is 1 o'clock. We'll give them just one more minute and then we'll get this started. Thanks for joining us today. So good afternoon, everyone. This is Diane Lechner with Midland Scientific. I'm our National Sales Manager. For those of you who were with us last week, thanks for joining us again. And if this is your first one, welcome to the FELC webinar series. So this is a two-part. Got the second part here on the USP potable grade alcohol production, and we will be focusing on the laboratory side today. So wanted to give a big thank you to all of the sponsors that are listed here, and thank you all for your time today. And also wanted to remind everyone, we do have all of the attendees muted for background noise, but if you have a question, we've got the Q&A section or the chat that you can use. We will wait until the end in the interest of time to answer all of those, but if you'd like to populate them, we'll make sure that we get to them. For those of you on the last one, we did run over a few minutes on some of those, but we'll make sure the entire session is recorded and we'll send that out here. So I'll go ahead and hand this over to Sean Van Hulsen, who will be moderating again for us today. Thank you.
Okay, thank you, Diane. Good afternoon, everyone, and welcome back. As Diane said, this is the second part of a two-part series on USP and potable grade alcohol production, part of a overall 4-series webinar that Midland is putting on in lieu of the face-to-face FELC this year. So several good topics coming up yet in the weeks ahead as well. So hopefully you can join for those. Without further ado, then we'll go ahead and get started to make the best use of our time and give that over to the expert speakers that we have here today. Our first speaker is going to talk about the methanol specification in the USP alcohol monographs. And that's Dr. Jenny Liu, who is a Senior Scientific Liaison in Global Science and Standards, excipients at the U.S. Pharmacopoeia Convention. She is currently responsible for development, updating, and harmonization of excipient monographs under the work plan of the Pharmacopoeia Discussion Group. She has more than 15 years' experience in analytical method development and validation for pharmaceutical products. She received her PhD in chemistry from Boston University. Supported by an NIH fellowship, she performed postdoctoral research in Susan L. Coleman Laboratory under Department of Chemical Biology, Ernest Mario School of Pharmacy in Rutgers University in New Jersey. So we welcome Dr. Jenny and look forward to her presentation. You can take it away, Jenny.
Thank you, Sean, for your introduction. It is a pleasure to speak about the most recent inclusion of methanol limit test in the USP alcohol monographs. Next slide, please. My colleague, Denita Broyles, introduced the USP to you in the last webinar. Our enduring mission is to improve global health through public standards and the related programs that help ensure the quality, safety, and the benefit of medicines and foods. And you can see the painting which showed our 11 founders of USP. Next slide, please. You may have heard about the methanol contamination issue in the alcohol-based hand sanitizers already. On July 30th, FDA sent a letter to USP requesting to include a limit of methanol test in the identification section of the alcohol monographs and any related USP NF monographs to help prevent methanol contamination. The methanol limit has already been required in the organic impurities test in the USP alcohol and the USP dehydrated alcohol monographs. You may wonder, why does FDA request an impurity test to be included as an identification test in the alcohol monograph? Actually, from a regulatory standpoint, it makes difference. If methanol detection and the quantification is part of the identification section, the CGMP regulations at 21 CFR 211.84D1 would require manufacturers of drug products to detect and quantify any methanol present for each lot of alcohol received. However, on the other hand, if methanol detection and quantification is only part of an impurity test, manufacturer need not include as part of its identity testing the detection and the quantification of methanol in alcohol. In addition, a manufacturer could deviate from the impurity requirements established in the monograph by labeling the product to indicate it deviates from the USP test requirements. So that's the reason. And next slide, please. On July 31st, USP reacted very promptly. We posted a notice of intent to revise. USP identified 6 related monographs in the USP NF and FCC, including USP alcohol and dehydrated alcohol monographs, USP isopropyl alcohol, and the USP azeotropic isopropyl alcohol monographs, and the 2 FCC monographs, ethyl alcohol and isopropyl alcohol monographs. The NITR was posted on July 31st to notify the stakeholders of the proposed revision of 2 monographs, USP alcohol and the USP dehydrated alcohol. Next slide, please. The NITR proposes to begin the revision process by first including the limit of methanol test in the ID section of the USP alcohol monographs. The added limit of methanol test refers to the organic impurity test in the USP alcohol monographs, and the acceptance criteria is the same as that organic impurities test as well, which is 200 microliter per liter. Please note, all 3 identification tests in the alcohol monographs are required to be in compliance with USP. Next, please. USP and FDA held joint stakeholder engagement calls with manufacturers, compounding pharmacies, and the similar facilities on August 10th to solicit feedback. Actually, this is a similar approach to the 2009 diethylene glycol contamination of glycerin products. So there was a precedent like this before, because alcohol, there are about 4,000 drug products contain alcohol or dehydrated alcohol as ingredients. FDA is concerned that this critical methanol contamination risk is poised to have a broad impact on the supply chain. Manufacturers should evaluate impact on their supply chain and work with FDA to prevent shortages. Next slide, please. USP implemented the US alcohol monograph revisions through an accelerated revision process, which is revision bulletins. You can see that the limit of methanol testing procedure and acceptance criteria are the same as that in the organic impurity test. And on the right side, there's a note there. This test must be performed to be in compliance with USP. So this is important. I re-emphasize it. Next slide, please. This slide is quite busy. Actually, I put a link there. So you can click the link and it links to the actual revision bulletin of USP alcohol monograph. This is the procedure of the organic impurities test. Please pay attention to two things. At first, the standard solution A is prepared using the alcohol sample as diluent. Many stakeholders missed that part. So they have misunderstanding on the acceptance criteria in the table 2 on the right side for the methanol, not more than 0.5, which is corresponding to 200 microliter per liter. Actually, not more than 0.05 has no unit. It is the result of which is the ratio between the peak area of methanol from sample to the peak area of methanol from standard solution. I will explain that later, and it's also included in our frequently asked questions. And the next slide, please. You may also notice that in the USP alcohol monograph, there are some diamond symbols. The reason is these two monographs are under the pharmacopoeia discussion group excipient work plan. PDG is represented by USP, EP, and the JP. PDG strives for the harmonization of monographs and the general chapters within these three regions. Both alcohol and dehydrated alcohol monographs are harmonized and have been official for many, many years, including the organic impurity test. So all the current revision to include the limit of methanol test is a USP local requirement. So it is indicated by the diamond symbols. USP has informed PDG about this revision, and the EP and the JP are assessing the issue of methanol contamination on their markets currently. Next slide, please. Here are the links to the revision bulletins. And very importantly, you can see the link to the frequently asked questions. You will find a lot of useful information there, such as why did the USP revise the alcohol monographs, why the limit test of methanol is included in the ID section, other changes applicable for alcohol used as API, etc. So I picked a few questions in the following slides. The next slide, please. So look at this one. So is USP methyl alcohol reference standard suitable for use in the limit of methanol and the organic impurities test? The answer is yes. Please note the reference standard name is USP methyl alcohol RS. So when you search the USP catalog, use this name instead of USP methanol RS. And this reference standard was evaluated by USP and found it suitable for use in both limit of methanol and organic impurities tests. Currently, the monograph does not require stakeholders to use these reference standards. However, our committee is considering to apply these reference standards in the monograph through a regular revision process, because due to the tight time for implementation, we don't want to have too many changes. You know, we will be using the regular revision process to implement these reference standards. And another question is, how does revision bulletins affect the harmonization of alcohol and dehydrated alcohol monographs? When you click the link PDG process, you will know more about PDG harmonization. So currently, the limit of methanol test is a local requirement. So it is only applicable for drugs intended for the US market. However, USP will follow up with PDG to discuss the opportunities to harmonize the ID section of these monographs with the inclusion of IDC. Next slide, please. So this is a quite busy slide, but it answers the question why and how methanol acceptance criteria not more than 0.5 correspond to 200 microliter per liter. So you can click the link to look at the answers. To explain this more explicitly using numbers, a table is shown here to demonstrate the acceptance criteria. Next slide, please. Yeah, next slide, please. So if you have trouble meeting the system suitability requirements, here are some tips for you. According to the USP chromatographic database, the G43 GC column from J&W Scientific was used for the validation of organic impurities test. You can use any brand of G43 column, of course. And the sample solution A is used as a solvent for standard solution B. So please check your chromatogram if there is any interference peak or baseline variation around the methanol peak. We received some questions about that. If you still have difficulties to meet the system suitability requirement, you may adjust the GC operating conditions according to the USP General Chapter 621. But please note, adjustments are permitted only when suitable standards are available for all compounds used in the suitability test. And these changes may require additional verification data. So next slide, please. The maximum variation of the parameters in 621 include column length, column inner diameter, film thickness, particle size for packed column, flow rate, injection volume, and the split volume, oven temperature, or the temperature program. So please check the chapter carefully for those parameters for your need. Next slide, please. Here are some important links to the hand sanitizers, including USP hand sanitizer toolkit, FDA guidance, WHO practical guide, TTB guidance, and the European Commission guidance, etc. Just for your information. Next slide, please. So if any access or potential drug shortage issues arise, please contact CDER at drugshortages@fda.hhs.gov. And if you have questions for the FDA regarding hand sanitizers, please email COVID-19 hand sanitizers at fda.hhs.gov. if you have questions about USP alcohol monographs, you can send your email to methanolid@usp.org, nfmonographs@usp.org. And if you have questions about FCC ethyl alcohol monographs, please email fcc@usp.org. So thank you very much for your attention. Yeah, we can answer the questions at the end of the meeting, the seminar.
Thank you very much, Dr. Liu, for that presentation and for the links as well. I think that would be very helpful to the audience. I believe these presentations will be made available to the group afterwards. As Dr. Liu said, if you have questions, if you could just kind of hold those or go ahead and put them in the chat box or the Q&A box now, and we will address them here at the end with our Q&A time. So the next two presenters, I believe, are presenting one fluid presentation here. They're both from Bion Sciences, so I'll go ahead and introduce both speakers here at the front and then turn it over to them for their presentation. Our first speaker is going to talk about analytical method development, process monitoring, and final product certification, and that's Tessa Schmitz. Tessa got her start in the industry with her undergraduate degree in chemistry and her master's in engineering management, both at the South Dakota School of Mines and Technology. She started her career at Bion in 2012 as the scientific services manager and loves to create analytics to support the ethanol industry here and abroad. In her spare time, Tessa can be found leading group fitness classes, hunting with her husband, and being a dog mom to their dogs, Scout and Sage. So we welcome Tessa and thank her for presenting today. And the second speaker from Bion Sciences will be Emily Reed. She's going to be talking about analytical standards for USP testing. To put it simply, Emily loves science. With an undergraduate degree in biology and an MBA, she stepped right out into the ethanol industry upon graduating and has been there ever since. In 2011, she joined Bion Sciences and has become the Director of Scientific Products. It is here where she produces standards and reference materials, all while striving to help customers have the most efficient and accurate labs and data. Outside of work, you can find Emily in the outdoors, spending time with her family and enjoying an ice-cold Diet Coke. So thank you, Emily, also for joining and presenting. And with that, I will turn it over to the two of you to go through your presentation.
Thanks, Sean. We'll go ahead and get started here. So as Sean mentioned, Emily and I are presenting to you from Bion Sciences this afternoon. Bion Sciences is a family of 3 companies. So there is Axion, as Sean mentioned, that's the analytical standards portion of the business that Emily heads up and is the director of. There is Soliton, which is the testing portion of the company, which is where I tend to spend most of my time. And then the last kind of third of the company is Comply, and they are the compliance branch. So they handle things like our PT programs, FSMA compliance, things of that nature. So collectively, we're very happy to serve sort of the entire breadth of the ethanol industry. Next slide, please. So we'll go ahead and get started talking about analytical testing for USP products. So we'll cover what you need to do those tests, what the tests are, and then we'll dig a little deeper into specifically the impurities, organic impurities testing. So right off the bat, it's a pretty simple list of equipment as far as lab testing goes. You'll need a UV vis, a hot plate or a water bath to do non-volatile residues, density and proof meter, a gas chromatogram is probably the largest investment you would have as you look to bring any of this testing into your own lab. And then just some consumables and reagents. You'll need some Nestler tubes, which are just a flat-bottomed test tube, graduated cylinders, beakers, volumetric flasks, and then there's a pretty short list of chemicals and reagents you'll need to purchase all of the organic impurities you're looking to quantify. There's some color and clarity solutions and chemicals that you would also need to bring in-house as you work towards doing any of these tests, if you want to do any oversight testing of this in your own facilities. Next slide, please. So here I tossed in just the full list of the test parameters and the related specifications. So up at the top of the chart, you'll see acetaldehyde and acetyl. That's reported as one test specification under the USP monograph. There's a spec for methanol, which Jenny touched on earlier. It's that 0.5 with no units, which corresponds to a 200 microliter per liter limit. Benzene, you have your total other impurities, non-volatile residue for those of you that do any sort of D4806 testing. That's very comparable to the gum method, the ASTM D381 that we're all pretty familiar with. There's a different spec limit on it, but it's the same vein of testing. There's a specification for specific gravity, 3 different UV vis measurements that are taken, and then clarity, acidity, and color of your ethanol solution. Acidity in this case is a bit more similar to the old acidity method that we're all used to as an industry, that ASTM D1613. Kind of the same, little bit different, but the same general idea applies. Next slide, please. So now that we've gotten sort of all the other tests out of the way, we're going to dive into specifically the organic impurities test. I know for lots of people in the industry, we've gotten lots of phone calls here at Soliton about it, and I know, as Jenny mentioned, there's been a lot of recalled product, and there's just some things swirling around kind of as it relates specifically to the organic impurities. So I'll spend the rest of the time really covering how you would do that in your facility lab, and a little bit about how it's different from GC work that you might already be doing. So the first thing when people call us and they have questions or they want to get into this testing, we always ask them, do you know your current impurity levels? Do you know how much fusel alcohol is in your ethanol? Do you know how much acetaldehyde you have in there? And for most people, if you haven't been looking at it, there's probably more than you think. So what I tend to prefer right off the bat is that you don't straight try and run the USP method as it's prescribed. These USP methods are designed to test industrial alcohol, which is very different than the fermentation alcohol that an ethanol plant makes. And so there's just some differences in the alcohol. And we've found that the USP method doesn't work very well if you already have quite a lot of impurity in your ethanol. So we in-house, we have a single injection direct measure method that we developed, and we actually developed it for the export industry of fuel grade alcohol out into those other markets. So we've had this method in-house for several years now. It's pretty quick, pretty easy. There's no spiking. You just run your sample, compare it to a known standard, and that will tell you, it's a very good starting point if you don't know where your alcohol currently sits as it relates to those impurities. It's also a little bit faster than the USP spiking method. So if you're working at your plant, you're working with like a contracted engineer firm, and you're working to make changes to your process, sort of clean up the impurities in that ethanol, the single injection direct measure method is going to be a lot faster. It's about a 45-minute run time, but you'll get results off. Like if you're making tweaks, it's a little bit more real-time measurement of those changes you're making to your process. And then as you clean up your ethanol, AKA remove the impurities, as you move down and get closer and closer and closer to those USP specifications that Jenny had in her slides, and it's table 2 in the alcohol monograph. As you move closer to those levels, then we start to suggest that you run the USP method. So that's going to be creating your 6 samples. You're going to have 6 injections on that GC, and you're doing that spiked injection process. Next slide, please. So the first thing that's maybe a little different than any GC work that you're currently doing in the lab is you're going to need to pick a different column. So some of you might be running ASTM D5501 in your labs now. That column will not work for this kind of analysis. You'll pick up some of the impurities, you won't find all of them. Some of them co-elude, you don't get good separation, you don't get a good recovery on a known standard, and those columns just really aren't designed for this kind of work. So what we recommend is the phase G43, which is what is spec'd in the USP monograph. So from our supplier, they happen to call that a DB624. That's just the name they gave it. What we had previously been running and would also suggest, if you're having any sort of column problems, is a DB1701 style column. That's what we developed our single injection method on, and we've had pretty good luck with that. It's a real robust column. We've actually just had to replace it a couple weeks ago, and we had used that column for about 3 years. So quite robust. They are a little bit of an investment. And again, if you don't have the option to do a dual injection, dual inlet, dual FID on your GC, it might mean that you're going to have to pull columns in and out more frequently, depending on how often you need to run this type of analysis. And then just a note about columns, every column and every column manufacturer, they might separate out the same components and they might be able to analyze the same thing, but be sure that you check the elution order on your individual column. I've seen columns from other manufacturers than we use, just a different style of packing or a different way they build those columns, and it can cause some shifts and some changes in the order that these impurities are going to come out in your detector. So just be sure that you know what you're looking for on your system. And it's best to do that with just straight injections of the compounds of interest. So you might not be able to purchase, 25 different compounds, but certainly purchase the big ones, the acetyl, the acetaldehyde, benzene, and methanol, and just do a straight injection of those compounds into your GC so you're really confident about retention time and the order that those are going to come off of your column. Next slide, please. And then we'll just touch briefly on instrument parameters. They are all listed in the monograph, and Jenny touched on them briefly. So for the oven parameters, there's not much that I recommend other than just the ramp that's listed in the USP parameter. The only caveat with that would be check your column manufacturer and the specific column that you end up purchasing to make sure that the upper end of your final temperature doesn't exceed the maximum operating temperature of that column, especially as you get into running. If you have the option to run D5501 and this USP method on the same instrument, you're going to have to be real mindful of that upper temperature range, because the D5501 columns can be a little hotter than the USP column recommendation, or at least the one we have here in-house. So just bear that in mind, because once you overcook your column, you're just going to have to buy a new column. There's no coming back from that. You can't trim off the bad part. If you overcook it, it's done. So just be mindful of that as you develop these methods internally. Split ratio is a good place to start if you're having problems with peak separation, detection, those sorts of things. It's usually the first place I go. It's a pretty simple thing to change in your software. Again, just look at your chromatography. Your chromatography will tell you a lot. The USP monograph, I think, recommends 20:1. I tend to run about 25 or 30:1. It's just what looks best on my system. But again, it can be very system specific, but it's an easy place to kind of make a change, make small tweaks, and just see how those tweaks affect your chromatography, whether you're making it more clear, more blurry, things like that. Carrier gas, they're early on in the sort of COVID-19 hand sanitizer rush, as we call it here. There was a lot of debate about helium versus hydrogen as a carrier gas. Some people had columns from different manufacturers that were recommending hydrogen as a carrier gas. I've never personally done that. We use helium here in-house. The USP monograph calls for helium, but certainly if that's something that your instrument manufacturer or your column manufacturer is recommending, I would certainly suggest following their recommendation on that. And then injection volume, 1 microliter is pretty standard. Again, if I have to tweak a method or go in and sort of change parameters around, I prefer to change the split ratio as opposed to the injection volume. So you pretty much always, across all the methods we run here in-house, I start with 1 microliter. That's a pretty easy place to start. And then again, I just adjust the split ratio till I get the separation and system requirements that the methods demand. Next slide, please. So with that, I'll turn it over to Emily and she'll talk a little bit about.
Hello, everyone. This is Emily Reed. Like the slide says, I'm the Director of Scientific Products and Compliance here at Bion Companies. And I, along with Tessa, am very happy to be presenting today. Next slide, please. So these are just a few of the things that I'm sure everyone has heard a time or two in their laboratories, from their coworkers, from your management team. We've heard it here at Bion as well. They see a data point, they don't agree with it, we think your instrument's wrong, there's no way that data can be right. What can you do in your laboratory, especially now with all the new USP testing that's going on? It's uncharted territory. You don't know what you should be looking at, what's normal for your ethanol. So what ways can you combat these topics from being brought up in your laboratories? Next slide, please. So how do we ensure the validity of our data? The first way that we would recommend is by the use of standards. These can definitely help show that your data is valid, that your instrumentation is working. So obviously every instrument, every method is a little bit different, but the standards allow for you to calibrate and validate your instrumentation. And one way we always say it too is we always bracket our samples just so we know during a run, if it's a longer run, we just know between all of these samples, nothing has happened. We know the samples, the first sample we ran that day and the last sample that we ran that day, everything is valid and all of the data looks good. And also to ensure the validity of our test methods, especially with the different USP, these may be new methods that you're running in your laboratory that you're not used to running. Those are able to help just to make sure you and your lab staff know that you're running the method correctly. Just a few other points. Another way that you can do the validity of your data is to do samples of known values. We have different proficiency programs or known value reference materials. And those two, it's always nice to know, yep, my instrumentation is running well, but also to be able, how is my lab, how is my data performing against other people in the industry, other labs in your fleet of your same company. It's just interesting to kind of know where you are compared to others, as well as a known value for that. Next slide, please. So kind of what Tessa had talked about earlier with the speciated GC method and standards, we had developed quite a few years ago, a standard that we had used for the export market, like she had talked about, to help validate the method that was produced in-house here at Bion. And what we have currently is a 20 component standard. And I just listed out a few of those components just because we've got a lot of calls, and those 3 components kind of seem to be what's forefront in everyone's mind and what they're most worried about quantifying in their laboratories. So once we started seeing the need for different standard levels, we have 5 different concentrations from 10 to 100 ppm. And we have also, a lot of times too, I'm sure you have all seen in your labs, if you've tested your ethanol for these different components, you can definitely start out a lot higher than 100 ppm. So we have had people who needed something 1500, 2000 ppm, obviously to, just to ensure I'm seeing everything correctly, I'm getting something at the top end and the bottom end of the range, just to make sure that, again, that data is right. And when you're presenting it, you know what you're presenting. We also done a few custom standards. The main custom standard concentration that we've been asked for an analyte is acetone. They are not, acetone is not in that 20 component standard, but it's a new component that has become of interest in the USP testing. So we have had some customers ask for a standard with that as well. The other, just a comment I want to make too about the lower range, it's always nice to get that lower range just because it's good to be vigilant on your chromatography at the lower end of your range. How Tessa just mentioned that we replaced a column, we started knowing that the column and the instrument was having issues because our 10 ppm standard that we were able to see great elution before, we were starting to be able to not see some peaks. So it's the standards, it's just a really good way to know what's the range that my instrument can even see. Just, instead of assuming, oh, we don't have anything in the ethanol. Well, maybe, or maybe it's just like Tessa said, you maybe need to tweak some stuff on your GC just to make sure that you're seeing everything correctly. Next slide, please. So just the conclusion, using standards and known value reference materials allow laboratory personnel to ensure the accuracy of their data, laboratory instrumentation, and methods. It's just a great way to know that everything is working smoothly the way it should be, and to be able to really have something to back up your data, especially when you're submitting it maybe to different regulatory bodies now. It's becoming a little more important maybe than it used to be, what data is coming out of your laboratory and where it's being sent, who it's being presented to. On behalf of Bion, we just want to thank everyone for listening today. We wanted to thank Midland for putting on this webinar series. We always really enjoy being at FELC and being able to be face-to-face with all of our customers. So even though we weren't able to be in Omaha this year, this is still a nice way to be able to connect with all of you. And hopefully next year we'll be able to be back in Omaha.
Okay, thank you, Emily and Tessa, for all that great information. This now leads us into our Q&A time, and we have ample time here to take in some questions. Diane, I think, did you typically handle the questions, or would you like me to?
You can go right ahead, Sean.
Okay. We have one here from Andy, and I'll go ahead and read this question from Andy. In my past history, it was common practice to qualify a small lot of an in-house material as a reference standard against USP standards, usually concurrent with the analytical method validation process. Has this process of qualifying an in-house standard remained generally acceptable? So it's a great question, Andy. I don't know if that is addressed to any particular speaker, but if there's one of them that have some experience or have heard something about what's going on out there, it seems like kind of a general question, I suppose, for maybe even some of the producers on the call, but I'll just pause here and see if anyone has something to contribute to Andy's question.
Yeah, this is Jenny from USP. So I worked in pharmaceutical companies previously. We also have, some in-house secondary reference standard qualification. But based on my experience, I mean, in both sides, both in USP and also pharmaceutical companies. So the USP standard, reference standards, are usually qualified by collaborative study by 2 or 3 labs. And also the reference standards are approved by the expert committee. Also the USP reference standard enforced by FDA. So generally when FDA audits a company, for USP, if USP standards are used, they generally have less questions. But of course, if you have your secondary in-house standard qualified, you'd better to show the full package to identify the structure, compound structure, the purity, the impurities. So all those documents are needed to prove your in-house reference standard are the actual compound and with the identity, quality, and the purity acceptable by the FDA. This is just my 2 cents. It's not to represent or on behalf of USP. So it's just my personal opinion on that. And also, I saw the other question, which is regarding in the Q&A section regarding using, replacing the helium with hydrogen. Thanks my colleague who responded mentioning the general notices 6.30 alternative procedures. So based on USP general notices, all alternative procedure needs to be fully validated according to USP General Chapter 1225, and must produce comparable results to the compendium method or procedure within allowable limits. So I just want to add a few points to that. Generally speaking, when you think about replacing helium with hydrogen as a carrier gas for GC, you'd better to think about, if the hydrogen will react with your analyte or not, because hydrogen might hydrogenate saturated hydrocarbons and aromatics. And also the safety precautions need to be applied because hydrogen has a broad flammability range from 4.0% to 74.5% in air. And also the performance. So usually if the same carrier gas pressure is applied with hydrogen as that with helium, the retention time may reduce, may be shorter. It may affect the resolution between analytes and the impurities in the system suitability. So that's, when you do the method development and try to change the carrier gas to hydrogen, you may think about these factors. And of course, your procedure must be validated according to General Notice 6.30. And another thing is the USP alcohol and the dehydrated alcohol monographs are under the PDG work plan for harmonization. No pharmacopoeia can unilaterally change the procedure without notifying other pharmacopoeias. So if we want to harmonize the procedure and change the parameters, we needed to get the agreement from EP and the JP as well. I think if there's a request for revision, please send the email to nfmonographs@usp.org, and my colleagues will, pass it to appropriate people, and we will work on that. Thank you.
Jenny, thank you for all that information. And I see there's some concurrent dialogue going on in the Q&A box as well as the chat box on some of these items. The folks from Bion, did you have anything just in addition to say about the carrier gas? And point well made, Jenny, that there needs to be equivalency established, but did you have a preference on carrier gas to pass on to the group?
I don't really have a preference. Every method that we currently run in-house uses helium. So when I developed the speciated method for the export market, I did it with helium because that's what we had, because that's what D5501 uses. So I don't have a preference other than my only experience has been with helium. But again, I would defer to whatever a specific method says or whatever an instrument manufacturer or column manufacturer would recommend. They know their products best, so that's really my only recommendation.
Hey,
Okay.
Sean, this is Christy. Could I respond to that?
Yep.
Yep, I agree with the Bion folks, and I'll just tell you this. My laboratory was so far inside the plant in Peoria that it was very difficult to move hydrogen helium cylinders all of the time. And so I actually went to a hydrogen gas generator and then proved equivalency. So whether I was running fuel samples or whether I was running USP alcohol samples, whether hydrous or anhydrous, I started using hydrogen gas as a carrier back in the 90s, probably circa 94, 95. And as long as you prove equivalence and follow the protocol, like Dr. Liu said, or on the ASTM side, right, you need to prove the equivalence on using an ASTM test method or standard practice, then I think that you're good to go. I personally did not see any issues with resolution or with the capillary columns. And I actually had a front and a back column in each GC, and they were each programmed with different timings, runs, carrier gas flows, etc. So it's totally doable. You just have to do the work, the due diligence.
Perfect. Thank you, Christy, and thank you to the Bion for the Bion response as well. A couple kind of related questions that have come in and look like they've been answered now. One was looking for the catalog number or the location to buy or procure the USP reference standards that were talked about, and that was answered by Jenny in the Q&A box. You can find it there. And then in the chat box, someone asked about the part number for the non-custom USP GC standards from Bion. That was also answered in that chat box, which I believe everyone can see, not just the panelists, the way it looks, but those part numbers were provided by Emily. And then there is one more open question here, I believe, again from Andy. This group might benefit from an overview of the analytical method validation. Could someone add their expertise beyond what we can read for ourselves? Maybe for Tessa or Jenny, anybody have any input there on the analytical method validation process?
I can give a little bit. It's a very broad topic, and kind of going back to what Christy just mentioned, it's all achievable, but you have to be willing to do the work and sort of put in the time, make the time to do these things. So I'll just discuss sort of how we handle it in-house, and certainly other people might do it differently. So what we tend to do is establish what you need the method to do. That would be the first thing. So we took in the case of the impurities, we took the impurity list that had been given to us by a customer for export market. So it contained fusels and aromatics and sort of all manner of jumbled up compounds that you wouldn't typically look for together. We took that to our instrument manufacturer and sort of asked them and what their recommendation would be, met with their column expert, got some baseline parameters, which if you don't have the ability to get the baseline parameters, that would, the bulk of your time and certainly a large portion of the R&D process would be setting appropriate parameters, finding an oven ramp that gives you good separation, kind of all of those nuts and bolts instrument things. So if you can start with, say, an application note from an instrument provider, something along that line that gives you sort of a rough roadmap, that would be greatly helpful. So take that application note, any of those things. From there, we did direct injections of all those compounds so that we could ensure that we had retention times, that we didn't have any colluting compounds. And then we slowly started to build up a standard of known concentration. So start with maybe 5 of the 20, add in a couple more. This all takes time, but you need to make sure that you, again, are seeing good separation. You're not seeing collution. You're getting the results you need to be getting. You have a pretty good recovery of a known concentration of things. So once we did all that sort of on the in-house level, we then purchased several third-party standards, did the same thing, injected them several times, calculated out recovery, made sure that we were seeing what we needed to see, getting the recoveries we needed to get on those components. And you just continue to do that until you have, in our case, probably, I think a good starting number is 25. Certainly the more data points, the better. But once you have 25 data points that you've sort of collected over time, so you're collecting maybe different technician error and error associated with different gas cylinders, because whether you're using helium or hydrogen, unless you have sort of like a generator or a really reliable source of that gas, you're going to see some fluctuations in instrument performance when you swap in and out gas cylinders. So maybe you want that error, you want maybe a different syringe in your auto sampler. You want to get a robust collection of data that sort of covers tip to tail of what you're expecting to see error in that instrument. Once you have that data set, then you can go through and start to apply some statistical parameters around that data set. Figure out your precision, figure out your bias, find the repeatability and reproducibility of your instrument. Maybe you want to take it a step further. If you have a sister plant or know of some other people working on these methods, send them a sample that you've analyzed so you can compare data and get that interlaboratory information flow going. There's proficiency programs that you can sign up for. You can like send Soliton a sample. We'd be happy to help anyone with that. As you're just going to start building a really robust collection of data. And the more data you have, the more confident you can be about that method. And the biggest thing I would stress with method development, specifically internal method development, where you don't have precision and bias and sort of all the nice endpoint information that something like an ASTM, like an consensus body method would provide you. The more data you have, the better that method is going to be. So that's sort of, in a very overview look, how you'd take something to say, I need to figure out how to measure X, and then how you develop like an endpoint method from that. I don't know if that answers the question or.
Thank you very much, Tessa. Yes, that was very helpful. It is the last question. There was one I wanted to come back to because I had missed it, and that was someone asked in the Q&A what the standard at Bion was produced in, and Emily answered that it's in ethanol. We also had someone in the chat box tell us that they couldn't see some of the things that were going on in the chat between the panelists, and so I had pushed some of that over there. You see my response to the question to Emily about what the part numbers were for their products. I threw that in there as well, and I see that Diane also forwarded that out to all panelists. It's just a little interesting here because they have a Q&A box and a chat box, and so some things are happening in both areas. But I think timely, it's the end of our 1 hour. There are no more questions in the chat box, and for the sake of time, we probably should close out the meeting for everyone. Diane, did you want to make mention of the upcoming webinars over the next couple of weeks that folks can look forward to?
Yep, you read my mind. So I just wanted to thank everyone for joining us today. Again, thank all the sponsors that are listed here, and for all of our panelists and moderator. We'll send a follow-up email with links to the presentations as well as the recording here. And then just a reminder to get registered for sessions 3 and 4. Next Tuesday on the 20th, we'll be discussing kernel fiber, so changing topic there. And then on the 27th, will be high protein feed technologies. So we've got some good panelists and speakers lined up for that, and hope you guys can join us for those. So thank you again for your time today. Hope everyone has a wonderful week.
Okay, thank you, Diane. See you, everyone. Hope to see you next week.
Yeah, thank you. Bye-bye.